Scientists tried to treat blindness for the first time in the world by using CRISPR technology and changing a patient's gene. Working at the Oregon Health and Science Institute in the US, the team dropped 3 drops of CRISPR DNA into a patient's eyeball. In this way, researchers hope to reverse Leber congenital amorosis, a rare genetic condition that causes blindness in early childhood. Charles Albright, head of the science Editas pharmaceutical companies, the Associated Press said speaks, originally take people who are blind, we can make them see. This may open the door to a completely new class of drugs that enter and replace your DNA. Quote, Leber's congenital amorosis, it is caused by a gene mutation that prevents the body from producing a protein necessary to convert light into brain signals. People with this disease often see little when they are born and may lose this ability even in a few years. Although conventional gene therapy can intervene in some genetic conditions by replacing the entire mutated gene rather than modifying it, patients with Leber's congenital amorosis were unlucky in this regard. Restricted viruses are used to transport genes into cells, but the gene that needs to be replaced is too large to fit in the virus. So doctors turn to CRISPR to edit the defective mutation. For this reason, they aim to replace or remove it by cutting both sides of the mutation. It is hoped that the ends of the DNA will be reconnected and allow the gene to function properly. The operation is performed under general anesthesia with a 1-hour surgery. Doctors, with the help of a tube with a hair thickness, they drip 3 drops of fluid containing the gene-regulating mechanism just below the retina, the lining of the back of the eye, containing the light-sensing cells. Eric Pierce, a doctor at Massachusetts Eye and Ear Hospital and working on the project, said, when the cell is organized, this is permanent. We hope that the cell will remain so for the lifetime of the patient because these cells do not divide. Quote, doctors think they need to correct 1/10 to 1/3 of these cells to restore vision. With this treatment, scientists have managed to correct half of these cells in animal tests. Doctors say that eye surgery is not very dangerous. Infections and bleeding, relatively rare complications. One of the biggest possible dangers of gene editing is that CRISPR can make undesirable changes in other genes. However, companies have done a lot to minimize this situation and ensure that the treatment only cuts the intended place, Pierce said. He advises Editas and helped test the Luxturna gene therapy, which is sold for a different type of hereditary blindness. Some independent experts are optimistic about the new study. He helped test Luxturna at Philadelphia Children's Hospital and was a researcher at the University of Pennsylvania. The gene editing approach is really exciting, says Jean Bennett. We need technology that can handle problems like these big genes. One day, he says, families seeking to cure hereditary blindness have sought him. This is a terrible disease, he says. They have nothing at the moment. Another gene editing expert at the University of Pennsylvania, Dr. Rental of Persons, according to experiments in human tissue, mice, and monkeys, this treatment seems to work. The gene editing tool remains in the eye and does not go to other parts of the body. If something goes wrong, there is little chance of harm, says Musunuru. This is a good first step for gene regulation in the body. In the new study, although CRISPR was used for the first time while editing a gene in the body, Sangamo Therapy, another company, is trying to interfere with metabolic diseases using the zinc-iron gene regulation method. Other scientists organize cells outside the body using CRISPR, trying to interfere with cancer, sickle cell and some other diseases. All these studies are different from the work of a Chinese scientist who was defeated in the international community in 2018. It was made explicitly with the approval of official regulators. He Junkui then tried to organize embryos using CRISPR and make them resistant to the AIDS virus. Unlike what is currently done to respond to diseases in adults, changes to the DNA of embryos can be passed on to future generations. According to the AP, it will be understood after about a month whether this first experiment worked or not. If successful, the research team plans to regulate the genes of 18 more patients, including children and adults. Doctors first used gene regulation within the body in 2017 with a different hereditary disease with the help of a tool called zinc finger. Many scientists believe that CRISPR is a much easier tool for positioning and cutting DNA at a certain point. So the interest in this new research is huge. Author, Marilyn Marchioni, Associated Press.